insights into the mechanism of resistance. Doctor, now the stage is yours.
Thank you very much. I hope that you can see the slides and you can hear me well. So my name is Lior David. I'm a professor of the Department of Animal Sciences of the Hebrew University of Jerusalem from Israel. And I know that most of the talks today are with the veterinary orientation. And I would like to discuss with you on something different, or in other words, how can we avoid the need to apply veterinary measures like treatments and vaccines as an alternative strategy to produce more fish and more healthy fish and reduce the stress of fish. So I will start with describing a little bit the importance of fisheries and especially of aquaculture. And what you can see in this slide is statistics about how much fish we produce by aquaculture as compared to how many tons of fish do we capture in capture fisheries by boats in the sea. And you can see that over time, the sector of aquaculture is growing rapidly. And it is actually the most rapidly growing sector producing protein from an animal source. Not only that, it is also a major protein source in many parts of the world where other sources are unavailable or more expensive. So eating fish and getting the protein need in our diets is very important, and aquaculture is providing this in many countries where other meats are too expensive or hard to produce. The good news is that while we, as you can see in the red graph, the capture fisheries, the fisheries in the sea, are more or less stagnant, and we cannot increase the amounts that we can fish in the sea, we can improve the production in aquaculture, and actually this becomes the most promising avenue to solve the need, the increasing need of human population for healthy food. And so it is very important to develop this sector and to increase the production of this sector. I will talk today about the common carp, about Cyprinus carpio, which is one of the most important fish in aquaculture. And as you can see in the slide here, the common carp, Cyprinus carpio, is among the most produced fin fish. And together with other cyprinids, as you can see here in the pie chart, it accounts for more than 50% of the aquaculture production. So really, in terms of the number of animals and in terms of the weight, production of cyprinids, among them the common carp, is by far the major animal in terms of numbers and production. And this brings with it a lot of challenges. And one of the most serious challenges for aquaculture in general, and as I said, I will give an example from the common carp, are infectious diseases. So infectious diseases, mostly viruses and bacteria, are the major threat to the progression of aquaculture, to the progression of the aquaculture sector. And it also a major factor for stress and reduced in revenue for the farmers. And in order to overcome diseases, there are several options. Some of them include prevention measures like vaccines, or trying to have more sanitization and prevention measures. In the case of bacterial diseases, sometimes we can use some medicines, but I myself, I'm not a veterinarian and I'm not a disease expert or practitioner. I'm a geneticist, so I would like to present today the third option to get sustainable aquaculture and to avoid the troubles that come with fish diseases. And this is the option of developing disease-resistant strains that even if the disease agent, the bacteria or the virus is present, they can actually either avoid becoming sick, or if they become sick, they will cure and recover and survive. And so there will be less damage to the farmers or even no damage to the farmer, and also reduces a lot the stress for the fish. So a little introduction on the common carp. So there are many common carp strains that are grown today in different parts of the world for food purposes, for production of food. And you can see some examples of food strains that are used in aquaculture. There are over a few hundred of different strains that are used in aquaculture, adapted to many, many conditions across the world for aquaculture. And besides the food fish, there is also this koi fish, which is the ornamental version of the same species. It's again the same species, Cyprinus carpio, but it was developed, or it was bred genetically to contain colors and color patterns and to serve as an ornamental fish. But essentially, these are the same species, and so the disease that I will be talking about apply for both food strains and ornamental strains. And you can see here an example of an outbreak of this disease. I will be talking on the disease that is caused by cyprinid herpes virus 3, which is a double-stranded DNA virus called CYHV3, or in other words, KHV, or koi herpes virus, named after the discovery of the disease in koi fish. And you can see an electron microscopy image of this virus here at the bottom, and some of the symptoms that you can see on fish as a result of infection. So this disease became very damaging across many parts of the world. And therefore, for several years now, my group is trying to resolve this problem by developing strains which are resistant to the disease, so that they won't be sick or they won't die from the disease. So if we would like to understand the resistance and this approach, then we have to make a distinction between resistance and survival. So obviously, survival of the fish in the face of a disease is the desired outcome. However, resistance is not equal survival. And you can see in this illustration, what do I mean? So in this illustration, you can see results of challenge experiments where we take fish and expose them to the virus. And we follow them throughout the disease challenge daily and count daily mortalities. So you can see on the y-axis the cumulative mortality by days. And in this experiment, what you can see is that we have in our hands fish that when we expose them to the disease will suffer high mortalities, so they will have low survival. As you can see in this graph on the side, most of the fish are dying, so less than 10% would survive. Whereas we also have fish that are already pretty much disease resistant, and those will suffer less mortalities and higher survival. As you can see here, they can reach to 90% survival, and only 10% would die from the disease. So in terms of survival, both cases, this graph on top and this graph on the bottom, will have some fish that survive. Here it will be only a small fraction, while here it will be a large fraction. but there will be survivors in both groups. However, there is a huge difference in terms of resistance between these two groups, because obviously in the group on top, most of the fish will die, and therefore most of the fish are not resistant to the disease. Whereas on the bottom type of fish, they are disease resistant. Okay, so what I'm trying to say here is that oftentimes people think that fish or any animal that survive a disease becomes resistant. So this is not the case. Resistant is only if this is genetically conditioned or genetically controlled, and so that the resistant fish will have resistance genes, and therefore also the progeny and the next generation of this fish will be more resistant to the disease than susceptible fish. Okay, and therefore our interest is to develop fish that are genetically resistant, that have higher chance to survive, and also will inherit this survival trait and the chances to resist the disease to their progeny. Also, I should say, because this is this meeting has a veterinary aspect to it, that we have to separate resistance from immunity. Obviously, individuals that are exposed to infectious agents like viruses will respond, their immune system will respond to the infection, and those that would survive will most likely be immunized, and therefore it will be hard to infect them, or it will be hard or easy for them to survive in a case where they will be exposed to the same pathogen for the second time. So I am not discussing immunity. Okay, I would like the fish, once they encounter the pathogen for the first time, to have higher or very high chances to survive, and rather than low chances to survive. So this is what I mean by genetic resistant, and now let's see some of the results. So first of all, when we started working on this, if you want to develop resistant fish, the first thing that you need to find is a source of resistance. You need to find some fish that genetically, that have genes that control resistance and will show high resistant levels. However, since the disease was discovered, it spread, as I said before, to actually all parts of the world where common carp is cultured. And this by itself tells us a very important piece of information, that actually all the strains and these different 100 strains that are used for aquaculture, they are all almost exclusively susceptible. Since we have outbreaks of the disease and reports on large damages to aquaculture ponds, this means that the fish are dying from the disease, means that they are susceptible and not resistant. So when the situation is like this, it is even more important to identify and develop by genetic breeding, develop resistant strains, because across the whole world, most of the strains, or almost all of the strains, are actually susceptible. And so in order to do so, I'm showing you that the different strains that we actually work with. We identified the strain which was more resistant, as opposed to the food strains that are used in Israel, which you can see here, which were susceptible like other strains across the world. So how do we see that they're actually resistant or susceptible? I will show you a few graphs like that. On the y-axis is, this time it's a survival level, the proportion of survival. And I'm showing you each dot as a representation of a family. So there are families with 100% survival, families with 80% survival, and so on and so forth. And here in the black bars are the averages of this particular strain. The strain was that had more resistant genes, whereas the fish that are used in aquaculture, including the ornamental fish, you can see that the average survival is very low. Okay, so now we have a challenge here. What is the challenge? The challenge is that the strain that is actually resistant is not suitable for aquaculture, for its growth rate, for its shape, for the scales that it has, the scale coverage that it has. The farmers won't grow this strain because it's unsuitable for aquaculture. So the resistant cannot be grown in aquaculture. It's not attractive to producers, whereas those that are attractive for producers and are used in aquaculture are susceptible. So we need to find a genetic solution to combine the two traits or the two situations to have strains that are suitable for aquaculture, but also resistant. And the way we have been doing this is by crosses and back crosses, by progression of resistance into susceptible strains. So this goes like this. We have the susceptible strain that is suitable for culture, and we have the feral strain which is unsuitable for culture but is resistant. We cross them to generate F1, and so we will have many F1 families, from which we will select the most resistant ones, and again cross them to the edible strain and generate many families and select the resistant ones, and then another generation, until we will get a strain that is both very similar to the edible strain and suitable for aquaculture, but also resistant. Okay. So I showed you what happens with the parental strains, the feral strain, which is unsuitable for aquaculture, versus the aquaculture strains, which are not resistant. But I'm showing you here on the right results from generations, from progressive generations, F1, and the next backcross generation 1 and backcross generation 2. And I am showing you this on top for one genetic background of an edible strain, and on the bottom for another background of an edible strain. So we've done that to two different strains so that we will see that it actually repeats itself in different genetic backgrounds. And what you can appreciate from this, comparing the parental strains to the advanced crosses, is that in the advanced crosses, we have on average the average of the parental strains, but we have a lot of variation around the mean. So in each of these backgrounds, we started seeing in progressive generations fish that are or families that contain fish which are very resistant, that we can continue and select. So this means that when we select resistant fish, we can transfer this resistance from the parental generation to the F1 and then to the backcross 1 and backcross 2. This is a good indication that we have a genetic resistance that we can actually transfer between generations. And now we have to continue doing this by selecting the best ones so that we will have eventually what we want, strains that are both suitable for aquaculture and resistant to the disease. And I'm showing you the third generation, and already in the third generation, they look much more similar to the commercial strains. So they are almost ready for farmers to adopt these strains. And as I showed you, they're also pretty much or much more resistant than the original susceptible strain. So on the side of developing these strains using genetic tools and selection, we are progressing, and this is something that we continue doing with the hope that we can actually produce strains that can be used in aquaculture pretty soon. And so besides this way to solve the problems of the producers, I'm also a scientist in the university, and I'm interested in genetics. And one of the most interesting questions is the genetics that is behind disease resistance. Not only because it is it is important commercially, but also because it usually involves the immune system. And I should remind you that the immune system of fish is very different compared to what we know about the immune system of other vertebrates, mainly land-based vertebrates. So by doing or studying resistance and seeing variation in the function of the immune system, we can study a lot on the immune system of fish, and this is knowledge that we actually need for various purposes. So what I would like to show you from now on are some results that relate to the mechanism of resistance. And the first one is when we saw the results of the feral strain versus the feral strain versus the commercial strain, one thing that popped to our eyes immediately was that the feral strain, which is resistant, has a full scale coverage over all the body, whereas the commercial strains, which are susceptible, they don't have almost no scales on the body. And this was selected many years ago so that it would be more more suitable for aquaculture. So we thought that this scale coverage across the body is serving as an integument that can protect the fish from infection and makes the feral fish more resistant than the commercial fish. And therefore, we actually tested this. So when we look across much of our data comparing strains which are mirrors, so they don't have scales, versus fish that do have scales, we see that on average, the scaled fish are more resistant than the ones that don't have scales, that are mirror. So we thought that scale coverage is an important protection mechanisms, but then we thought about it again, and we actually saw that the reason we see this difference is because the scaled fish were more related to the feral strain, which is also scaled, and therefore the feral strain brings the resistance, and this is what is causing this difference. And in order to actually control the difference in the genetic background and ask whether the scale pattern determines resistance, we have to do it on progeny from the same generation and even from the same family, so that they will be genetically very similar, except for this trait of scale pattern. So we took fish from the same families, brothers and sisters. In some of the Becros-1 families, we had families where half of the fish were with scales and half of the fish were without scales. And we compare actually the survival rate or the resistance of these fish on the same genetic background of the same family, brothers and sisters. And what you can see here now is that there is no difference between the means. Each of these two dots are the scaled fish and the mirrors, and without scaled fish and connected by a line are fish from the same family. And so you can see that on average, there is no difference between brothers and sisters despite this difference in the scale pattern. So the scale pattern is actually not helping the disease resistance. And this is good because, as I told you before, for aquaculture, we want fish without scales. And so this means we can generate fish which are without scales, but highly disease resistant. So another question that we ask ourselves is, what is the nature of this resistance? So what happens when we infect the fish with the disease, with the virus, and how, what is the difference between the resistant fish and the susceptible fish when we infect them? So what you can see here is the experimental design. We use fish from a susceptible family versus fish from a resistant family. Time point 0 is the time when we infected the fish with the virus. And then we took samples of tissues and extracted the DNA at different time points from these two types of fish. And using qRT-PCR, we compared the viral load in the tissues as a function of time. So the results were as follows. Here in this graph, you can see the blue line, and the right scale is the actual mortality in the experiment. Okay, so we have resistant and susceptible. So overall, it's around 50% mortality in the experiment. But what it also shows us is when the disease becomes more serious, day 0 is the infection, and we start seeing mortalities around day 6 or 7. Okay, so we measured in the tissues the level of virus. And what you can see in the red points are the levels of the tissues of susceptible fish, and in the green dots, the levels in resistant fish. And you can see that as of day 4, we see significant differences between susceptible and resistant fish in the viral load. The susceptible fish will have much higher viral loads. This Y scale here that describes the viral load is a logarithmic viral scale. So you can see that in day 8, we have about 100 times more virus in tissues of susceptible fish versus resistant fish. So we can see that across all tissues, but we can see it also for specific tissues. Almost in all tissues, we found the same result, where the resistant fish would have, the green ones would have much lower viral load than the susceptible fish. So the conclusion from that is that the mechanism of resistance is depending on the capacity, on the ability of the resistant fish to limit the progression of the disease or the replication of the virus, and it actually control the viral load to a level that the fish survive, whereas susceptible fish lose control over the virus, and that's the reason why they actually die. So this was very important because that indicated to us that the resistant fish has a mechanism to control the viral replication, which is most likely the immune system of the fish. So the next thing that we did is we wanted to see whether we can identify some genomic regions or even some genes that control resistance. So find what are the genes that contribute to the resistance mechanism, and also find markers so that we can use these markers to select for resistant fish. And so we did the following experiments. These are different families, and you can see here the mortality curves by day. And so from each of these families, we sampled fish that were actually dying early from this time point. And also after the mortality stopped, we sampled fish that survived the infection. And we compared the DNA of these two groups. And when we compared the DNA of these two groups, we identified several regions in the genome that actually control resistance, the difference between those that died and those that survived. What you can see in these graphs, briefly, in all these different colors, you see the different chromosomes of the common car. Each color is a different chromosome. Each of these dots is a polymorphism, is a DNA marker that we actually investigated in all these fish. And on the y-axis, you can see the significance of the tests that differentiate the susceptible from the resistant fish. So when the points are up high, that means that in this part of the genome, there is a huge difference in the genotype, in the DNA sequence between susceptible and resistant fish. So we identified, we did this actually in 3 families, and I'm showing you here only 2. And we identified 2 major regions in the genome, this one, and we call them QTLs, quantitative trait loci, number 1 and number 2. Number 2 we found in both families. Number 1 had a significant effect in this family, but a lower effect in this family. But and this added up to a previous study that we actually identify another region in the genome that contains the gene IL-10, interleukin-10, that also contributes to the resistance. So we were very happy by these findings because of 2 reasons. Why? One is that now we can select some DNA markers here that can help us identify, molecularly identify, genetically identify the resistant fish, and not just relying on disease survival, or actually relying on both, not just one. And the other one is that this is a very good proof that there are genes that contribute to resistance, that the resistance that we are developing has a genetic basis to it, and we actually are developing genetically resistant fish. So we continued with identifying such genes and places in the genome and markers by another experiment where now we had 9 different families that were variable in the survival level, as you can see here in this graph, altogether about 300 fish from 9 different families. And again, we use the method based on next generation to genotype these fish, a method called genotyping by sequencing, and we actually work with about 60,000 DNA differences in the genome for all these 300 fish. And we looked for the association between DNA genotypes and survival and resistance. And again, I'm showing you the same type of plot, where again, high level dots are markers marking regions of the genome and genes that actually contribute, that control disease resistance. And so in the second experiment, you can see that we identified in this family three different regions in the genome. One of them is the same region that we identified previously. So we were able to repeat this result, confirming this result, validating this result, which is again very important to believe that this result is actually true, but also other places. So we had this QTL1 from the previous study, which we didn't identify in the second study, but we had also this site and this site that we did not identify in the previous study, which is good, meaning that we discovered more regions in the genome and more genes that actually control disease resistance. And we also did another analysis where we actually compared the differences in percent survival. As you saw before, these families are not just having each one fish that survived and died, but also different levels of survival. So we compared the families that had high survival versus families that had low survival percentage, and identified 5 more regions in the genome that actually control family survival. So altogether, we identified close to 10 places in the genome, 10 QTLs, and the genes in these QTLs that contribute to disease resistance. And so we also tested them across 71 individuals, which are unrelated, coming from separate families that we have in our hands, to show that actually many of these places actually verified also when we were studying the population. So these are markers, these are regions that we can actually use to select for better resistance based on the genetics, not only based on the phenotype of survival. And these results were already published in these 2 papers and the papers that I showed you, that I cited before. But what this result tells us is that actually there are many places in the genome, or several places in the genome, that there are genes in these places that control disease resistance, which means that this disease resistance from the genetic perspective is a polygenic trait. It has many genes that control this resistance, which is not often found for disease resistance in other fish and other diseases or in other animals, where it's most of the times a single gene that controls resistance. Here it's a polygenic trait and it's much more complicated to work with, but for me it's more interesting and more challenging. Okay, so the last section that I would like to discuss with you with respect to the mechanisms of resistance is when we compare the transcriptomes of these resistant and susceptible fish. So we actually used RNA sequencing to sequence the mRNAs for these susceptible and resistant fish and compare them to actually see which genes are differentially expressed between susceptible and resistant fish. And again, we did this experiment by comparing these two groups and sampling different time points before we infected the fish and after we infected the fish. And the results that I'm showing you today is the comparison between the before infection and day 4 after infection. You can see here the graph of the mortalities. So day 4 is before the fish start mortalities. So it's relatively early after infection. So the first result is presented in this Venn diagram. So we actually had 4 different treatments that are labeled here as follows. R stands for resistance, S stands for susceptible, 0 for day 0 before infection, and 4 for day 4, which is post-infection. So we could do and identify genes that are differentially expressed between any couple of treatments as shown above. So basically 4 combinations comparing the differences in gene expression between susceptible and resistant fish before infection, between susceptible and resistant fish at day 4 after infection, and also between susceptible fish after infection to before infection of susceptible fish, and after infection to before infection of resistant fish. And you can see in this Venn diagram the overlap between these 4 comparisons, showing that actually we identified few hundreds of differentially expressed genes, but they are not overlapping these different comparisons, okay? So to focus more on the mechanism of resistance, what we actually wanted to see is the response to infection, is the comparison between day 4 and day 0. So between day 4 and day 0, we did the comparison for both the susceptible fish and the resistant fish. And what you can see are the susceptible fish here in pink and the resistant fish in blue. And the overlap in differentially expressed genes in genes that are upregulated, that the infection induces their expression, versus the genes that were downregulated, so the infection suppresses the expression of the genes. And to make a long story short, what you can see are 2 important points. One is that the circles for the susceptible fish, the pink circles, are much larger. So in susceptible fish, there was a much more dramatic gene expression response than in the resistant fish. They respond to the infection much more significantly, changing levels of expression for many more genes than the resistant fish. So the resistant fish, the infection did not make a significant difference in their physiology, in their gene expression. The second important part is when you look at the overlap between the susceptible and the resistant fish, you see that most of the genes are specific to either resistant or susceptible fish. The overlap is relatively small, which means that the response to infection was significantly different in resistant than susceptible fish. And this was nice because we wanted to capture the difference between resistant and susceptible fish to understand the mechanism of resistance. And so the next step was taking these differentially expressed genes in susceptible separately from resistant, in susceptible in red and resistant in green. And I'm showing you here the enrichment or the significance of genes that belong to these different functional categories, gene function categories. And what you can see that in these, that the genes that we actually identified as differentially expressed belong to categories which were very encouraging, like defense response, immune system process, leukocyte migration, response to virus, and so on and so forth. So most of the genes that we identified were actually part of the immune system, which means that, again, according to our hypothesis, based on the load of the virus, the resistant fish are different from the susceptible fish in their immune system, in their immune response. They respond differently immunologically than susceptible fish. So there are many fish for those of you who knows, but I will focus now on a couple of main differences. One is that we saw in susceptible fish a typical interferon viral response. So interferon and interferon-induced genes were highly expressed in susceptible fish, but not in resistant fish. And we also saw differences in the repertoire of chemokines, which are signals for different adaptive immunity mechanisms. So chemokines is a very large family of molecules, of proteins, that we can distinguish into different families like CCL19, CCL38, CCL8, 18, and so on and so forth. And for each of these families, we can see here that there are multiple copies in the genomes of fish in general, and also in the genome of the common carp, the zebrafish, and other fish. Okay, so this is a very complicated genetic basis to study, because it has many families, and each of these families have different functions, and within each of these families there are different members that can be differentially expressed or not. You can see on this phylogenetic representation the genes that were actually differentially expressed that are marked by these asterisks. So you can see that within each of these families, some of the members are actually responding to the infection, whereas others are not responding to the infection. And these are all, for instance, CXCL8 member families. Okay? So the diversity is very large, and our experiment was very sensitive in capturing the specific and subsets of genes that actually responded to the disease and were part of the resistance mechanism. And when we looked at the differences between these cytokine signals, the differences between resistant and susceptible fish, we actually saw that the signals, the cytokines that are induced by resistant fish, belong to neutrophil attraction. So they are the chemokines that activates and recruit neutrophils to defense against the infection. And neutrophils are usually not participating in viral infections, but for fish, as I told you, there are differences in the immune system, and probably in this example, either the neutrophils are part of the immune response to the viral infection, or the cytokines are not signaling neutrophils as they signal in mammals, but they are signaling something else. So we don't know this yet. So resistant are much more focused on activating the neutrophil signals and the neutrophils to protect against the virus, whereas the susceptible ones are activating leukocytes and mainly macrophages, which is more typical to a viral infection. So we see in general that the susceptible fish are more typical in their immune response. They first respond by eliciting the interferon and interferon responsive genes pathways. And you can see specific real-time PCRs to confirm some of the most important markers of the interferon pathway, like VIP1 and MXC. And indeed, you can see that they were elicited differently between resistant and susceptible fish. And also some of the chemokines from the CCL19 family that were different in susceptible fish versus resistant fish. So we were able to confirm the activation of this pathway, which attract leukocytes and macrophages. Whereas in susceptible fish, but not in resistant fish. Whereas in resistant fish, we were able to verify the activation of the pathway that induces a neutrophil response, as you can see here by this example of CXCL8, different copies of this gene that was activated in resistant fish, but less activated in susceptible fish. So in summary, what I showed you today with respect to so disease resistance is a very good strategy to avoid the damages of infectious diseases in aquaculture, and also to reduce the stress from disease of fish, because we have very limited other control measures. And what I've shown you today is that with respect to carp and to this disease, we identified a polygenic disease resistance. So the basis is polygenic, many genes, and I showed you some evidence that actually the difference in the mechanism relates to the immune system. So resistant fish get infected but control better the virus replication, the viral load. And they also elicit a very distinct transcriptomic response to the infection that include a lot of genes, but essentially the susceptible fish are eliciting the typical interferon response, whereas the resistant fish do not. And in terms of the cellular immunity, the susceptible fish attract macrophages and T cells, whereas the resistant fish are more attracting neutrophils. So they are using different arms of the cellular immunity that leads to a different result, to resistance or susceptibility. And this is very encouraging for us, because if it is the immune system that functions better in the resistant fish versus susceptible fish, then maybe the immune system will also function better with respect to other pathogens, which means that resistant fish will have better immunity, more general better immunity against multiple pathogens. And so therefore we will not have to develop separate resistant fish to future diseases, to other diseases. And with that, I would like to end and thank my group members, several students and the PhD students and postdoctorals that helped in this project throughout the years, and also some of my collaborators from Israel, from the US, and from the Netherlands, and from the UK, and also a company in Israel that is we work together with to develop vaccines to this disease. And so thank you to all these that supported this result, and thank you for your attention, and I will be happy to take questions if there are any.
Thank you for that insightful talk, Doctor. Now the stage is open for questions. I assume that you have no questions from the speakers. Once again, I thank you for being a part of this webinar, Doctor, and hope to associate with our future events. Thank you.